Yes, I'm sure you've all been on the edge of your seats waiting for my title. Yeah, so what happened is a speaker wasn't able to come. So we thought that this would be a great time to insert this topic into the exciting discussion that's already going on at the conference. So disclosure, this is not actually my work. This is actually the work of Camilla Bond. She was our phenomenal MD PhD student we had that graduated. She's now doing her neurosurgery residency at UPenn. Definitely look for her doing great things in the future. But yeah, basically I'm the spokesperson today. So I am early on in the proposed author list, so I should know what I'm talking about. Is this the clicker? All right, so we're in Kristen Swanson's lab, and as many of you probably know, we work with glioblastoma. And glioblastoma is a primary brain tumor known for wide inter and intra-patient heterogeneity. It's very aggressive, but clinically, what's going on, like in the clinic? So the clinician, because of that very eloquent location of this tumor, they're not able to really have a lot of access to tissue. So pretty much through the entire course of the disease, they're really working with imaging. And this is basically what they're seeing. So on the left is a T1 GAD image, on the right is a T2 flare. The T1 GAD is basically showing them where the blood-brain barrier has broken down, and the T2 flare is basically showing them where there's extra fluid. And this fluid could just be fluid, it could be immune cells, it could be cancer cells, but they're not really sure. So how do they make decisions in the clinic? Well, what usually happens is when they go to surgery, there is a single biopsy taken from this region. And from this biopsy, they are going to make their diagnosis. So that involves like immunohistochemistry assessments. They'll probably do a little bit of genetics. They might do some RNA-seq, but basically they're going to take this one biopsy and have to make extrapolations for the entire tumor. So they have to think of it almost as homogeneous when they're making their decisions. Now, there are a few cases so when they're thinking about cell density, they can make some educated guesses. So they think about like the T1 GAD is probably a higher cell density, the T2 maybe lower cell density, and that can help them in thinking about surgery targets and radiation targets, but they're still educated guessing and they're not really sure, and this is only on cell density. Of course, the truth is going to be much more complicated. And here I'm only showing 2 colors, but as all of your presentations, is probably more like 4 to 10 or who knows how many colors, right? So in the clinic, I'm specifically going to be talking about how this influences how they're dealing with immunotherapy. So immunotherapy is just a whole other beast because imaging is already complicated and immunotherapy takes it just to the next level, right? So here is an image of time sequence of a patient on immunotherapy. So the number there is indicating the month relative to the therapy. So -1 means we're before surgery, then 047, we're moving on. You can see on this T1 GAD image that abnormality is getting bigger. But somehow at month 7, 9, 11, it looks like it's stabling out, and then it starts shrinking. So like I said, when we're looking at these images, we don't actually know what we're looking at. What we can infer from this is that it was growth that we're probably visualizing actually an immune response that is looking like tumor recurrence, but then it's ultimately resolving itself. Now, the issue is that glioblastoma patients do not generally live very long. The median survival is only 15 months. And the clinical recommendation is to make your IRANO assessments or resist or whatever at 6 months with immunotherapy. So you're looking at this chart here at 6 months, what's your call going to be? It's very unclear, right? So imaging is confusing, it's non-specific, and generally it just leaves more questions than answers. So is there something that we can do to make this a little bit better? And what will that take? That's what I'm going to be talking about, one of our efforts. So I think I went too far, right? So we have been thinking like, how do you get at this? You kind of need to have more samples, right? You need to know more about what you're looking at. And over the last 5 years, I'm not going to spend too much time on this because there's a whole talk tomorrow on it, but we have been collecting a really fantastic set of data of image localized biopsies. So what this means is that the surgeon is taking tissue from the patient, and we actually know approximately where on the image is coming from. And this is really exciting. We're getting more than 1 per patient. So on average, we're actually getting about 4. And yeah, so we're seeing what we can do with that. Of course, our big vision, the big dream is that basically through this, we're going to be able to connect the biology to what we're seeing on the imaging, right? So the idea is you have your biopsies, then we have all this different clinical imaging that's available for these patients on our trial. you're able to figure out like, whatever biology you're interested in, histology, genomics, transcriptomics, and then connect it to the regional imaging features through the power of modeling, machine learning. We're hoping then that we can train a model to give you the regional imaging, information about the underlying biology from those images. So that's our goal. Now, in this talk, since we're looking at immunotherapy, we're specifically going to be hoping to model where T cells are. So we're going to be looking at transcriptomics. Now, the thing is with these biopsies, they're, when you do any sort of analysis with the tissue, it's destructive. So you can really only do one. If you can cut it in half, you can do a couple things. But so what we generally do with them is we do RNA sequencing, bulk RNA sequencing. But I want to know about T cells. How do we get down to just the subpopulation and really thinking about that? Well, we have been using something called CyberSort. Another version is CyberSort X to deconvolve these cell populations. And just real quickly, the general idea is that this whole algorithm is trained ahead of time with, you have some single cell data, single cell RNA sequencing, and you get some idea. Each of the cells have been tagged as a type of cell that you think is going to be in your sample. And then you get an idea of the transcriptome profile for that cell type. You can then create a gene expression signature matrix that you think, ideally, if you were to multiply that by a vector, that's the percentage of those cell types in your sample, you would ultimately get to realize your bulk RNA sample. So basically, this is looking at an AX equals B kind of situation. So it's an inverse problem. It is overdetermined, very much so, but you can do some regression and you can get an estimate of the cell populations that are in that sample. So that is what we've been doing. The library that we were using for the single cell was a default library in the CyberSort. It was leukocyte, so I think is pretty focused on the hematopoietic system. But it breaks it down into 22 cell populations. 7 of them are different T cell subpopulations. So when I talk about T cells later, I am going to be talking about the sum of all of those T cells. All right, so just recap. What am I trying to do? Because there's a lot going on here. All right, we have MRI localized biopsies. That means we have a biopsy, we know where it is in the brain. So from the imaging part up on the top, we think about where it is at each of those spots for the biopsy. We are going to be calculating, figuring out a lot of the features there. So feature extraction, there will be feature selection there. On the bottom, we also have tissue, and we are doing RNA sequencing and then deconvolving it into the different populations. And then we are doing a radiomics model, in this case, an SVM regression for predicting the T cell population at each voxel or at each biopsy. So how did we do? I'm going to go ahead and say we did pretty good. So this data set, we had 147 biopsies. You might think that's a lot. You might think that's a little for machine learning, but that's what we had. 81 were male, 66 were female. As you just heard, we have a great collaboration with Josh Rubin, and we are very focused on sex differences. And we do think that there is reason that the imaging and, well, the biology would likely be different, and thus the imaging to biology would be different. So we did train male and female models separately. Anyway, the top is our training data. And as you can see, the x-axis is the predicted, the y is the actual, and we're doing pretty good job hitting that identity line. So we're really excited about that. The test data is still also doing pretty well. So I would call this a model that has hope, right? But any model could have hope at this stage. But the real question is it functionally useful? When you take it to the clinic, does it mean anything, right? So we were fortunate enough to be working with a clinician at Mayo Clinic who had this phase 1 dendritic cell vaccine trial where there were 20 patients on it, and the trial came back as negative. So that means on average in this 20 patient set, there was no improvement in overall survival. But, with every single clinical trial, there is the question, was there a patient we missed that actually got benefit? Is there a better way to think about who should have been on the trial in the first place? And so we were thinking, if we applied our model to this, is there something like that we could find and figure out? So that's what we did. So we took each of those patients. We had a pre-treatment image and we had a post-treatment image. We segmented out the tumor regions on both of those. We then applied our model on this, and then we could make our prediction for each voxel, how many T cells we think were there. And for this initial pass, I will say that we weren't doing very spatial, like voxel to voxel comparison. We instead were doing like a sum of all the T cells pre-treatment, a sum of all the T cells post-treatment, and we were looking at the variation between that. So for this specific one, we were also focused on T1 GAD and T2 flare images only. That first slide before had a few other images, but these are the two that are available pretty standardly clinically. And that is also really important for these kind of models. So what we were wondering is if we look at it, like if it goes up or down in the T1 GAD or up or down in the T2, what do we see? And what we found was that if you think about that, like that kind of quadrant over there, that if you are in the situation where your T cell population was predicted to go down or be stable in the T1 GAD, but go up in the T2 or flare, then we were actually seeing a differentiation in the survival of the patients of those two cohorts, if you were in that quadrant or if you were not. And in fact, in a Cox proportional hazard regression analysis, that was the only thing that came up as significant in this survival analysis. So I would say that this is very exciting, tantalizing data, but of course there's always more work. Yeah, so in summary, interpreting clinical imaging is challenging. Just period. We have trained a model to predict T cell abundance based on the clinically available imaging, and it does seem to have some discriminatory ability. But the future work, always more data, right? So we trained this on 147 biopsies. We actually did recently get another 50-ish analyzed with bulk on RNA-seq. We're hoping to get even more than that. Hoping to get more funding for some of that. And we also have connections with a few more immunotherapy trials and their imaging protocols so that we're hopefully going to apply it there and see if it is more broadly deployable and interesting. So with that, thank you, of course, to all of our funding for this. Lots of NCI support, lots of James McDonald support also. And then this is our team. And again, just really want to point out, this is Cam Bond's work. She is amazing. She does great things. And thank you. Any questions?
Yeah, that indeed is amazing. Have you considered using imaging-based flow cytometry to identify T cell presence rather than the RNA-seq approach? Because that would be much faster, and I think the technology is available now. So,
it would be much faster?
So you can do this in real time, right? Oh. So you take the biopsy, and within 15 minutes, you know exactly how many T cells you had where. So tomorrow during the talk where they're talking about when we collect all the data, you'll get to hear about all of the challenges of in the OR room, what's going on. Yeah, awesome. So, I mean, yes, I would say that we're always open to discussions of how to make that faster, you know, quicker, better, faster. But there are logistical challenges in the OR room. I can only imagine.
Yeah.
Super cool.
Okay. Thank you.